Today, we're going to learn about transport across the cell membrane in the easiest way possible. Basically, this lesson is all about how things move in and out of the cell. You might be wondering, when am I actually going to use this? Unlike topics that teach a specific skill like cooking, makeup, or driving, this lesson is more about understanding how living cells work. It is a foundation topic that becomes especially important for people who want to become doctors, nurses, medical laboratory scientists, pharmacists, vets, biologists, chemists, agricultural scientists, researchers, and many more. So let's begin. First, organelles are tiny parts inside the cell that each have their own jobs to keep the cell alive. Examples of organelles include the nucleus, the cell's control center, and the mitochondria, the cell's energy producer. Now, the main focus of this lesson is the cell membrane. The cell membrane is like the cell's gate or wall because it controls what can enter and what must leave. Before we talk about movement, let's first understand what the membrane is made of. The cell membrane is made of phospholipid bilayer. A phospholipid bilayer is simply two layers of tiny fat-like molecules stacked together, kind of like a double wall surrounding the cell. The membrane has an oily middle layer, where substances like oxygen and carbon dioxide can pass through easily. However, substances that mix well with water, such as glucose and ions, usually need protein channels or carriers to help them move across the cell membrane. These membrane proteins help move materials and receive messages. Meanwhile, carbohydrates in the membrane, found as glycoproteins and glycolipids, which are molecules with small sugar chains, act like name tags so cells can recognize each other. At the same time, cholesterol helps keep the membrane from becoming too soft or too hard. Now that we know the parts, let's look at what each one actually does. Proteins are molecules made from the protein we get from foods like chicken, fish, eggs, and beans. There are many different kinds of proteins in the body, and each one has a different job. Fun fact, the proteins you eat don't stay exactly the same in your body. Instead, your body breaks them down into smaller pieces called amino acids, then uses those amino acids to build the proteins it needs. For example, some proteins help reactions happen faster, some help to receive the signals, and some help transport materials. We're going to talk about later what are those. Meanwhile, glycoproteins and glycolipids help cells identify and communicate with each other. On the other hand, cholesterol helps the membrane stay flexible and stable. So overall, every part of the membrane has an important job. Now, let's move on to the fluid mosaic model. The fluid mosaic model explains the structure and composition of the cell membrane. Let's break the name down. In here, fluid means a substance that can flow, like water. In the cell membrane, it means the membrane can move and stay flexible. Mosaic means it is made of different parts working together. Model is a scientific presentation of how something works. So, the fluid mosaic model describes the cell membrane as a flexible structure made of many moving parts working together. Instead of being rigid, the membrane can move and adjust when needed. To help the cell membrane perform its many functions, it contains different types of membrane proteins. Let's take a look at them. Membrane proteins are special proteins attached to the cell membrane. They help the cell transport substances, receive signals, speed up reactions, and provide support. Integral proteins help support and stabilizes the membrane, like a table that keeps things steady and strong. Pump proteins use energy to move substances across the membrane, like a balloon pump pushing air inside a balloon. Channel proteins act like tunnels that allow substances to pass through, like a tunnel under a bridge that lets cars travel from one side to another. Carrier proteins move specific molecules, like a taxi picking up one passenger and bringing them to a destination. Enzyme proteins speed up reactions, like a blender quickly mixing ingredients. Receptor proteins receive chemical signals from outside the cell, like a bell that rings when someone pulls it. Glycoproteins help cells recognize and identify each other, like a name tag that tells who someone is. Anchoring proteins hold the membrane in place and connect it to the cell's internal structure, like an anchor that helps the boat from drifting away. Each protein has a specific role in helping the membrane function properly. Now, let's look at how substances move across the membrane. Before we continue, remember that membrane proteins and transport mechanisms are not the same thing. Membrane proteins are the structures that help substances move across the cell membrane. In contrast, transport mechanisms are the names of how substances move. So don't be confused if some of the drawings look similar. The proteins are the structures doing the work, while the transport mechanisms describe the way the movement happens. Transport mechanisms are the different ways substances move in and out of the cell. Transport means moving something from one place to another. Mechanisms means a method or way something happens. First, let's talk about passive transport, which is the movement of substances across the cell membrane without using energy, or the ATP. Simple diffusion, facilitated transport, osmosis, and filtration are all types of passive transport. Simple diffusion is when small molecules move from a crowded area to a less crowded area without using energy, like the smell of perfume spreading across the room. It's move where there's a lot of perfume smell to where there's less. Facilitated transport works the same way, but molecules need protein helpers to cross the membrane, like entering a building using a door. The molecules cannot pass through the wall by itself, so it uses a protein or the door to get across. Osmosis is the movement of water across a selectively permeable membrane from an area with more water to an area with less water. Like a dry sponge soaking up water, water moves to where there's less. Filtration is the movement of water and small dissolved substances through a membrane due to pressure, like using a coffee filter. Pressure pushes water and tiny particles through the filter while larger particles stay behind. All of these are passive transport, which means they do not need ATP and move naturally. Unlike passive transport, active transport needs energy or ATP from a less crowded area to a more crowded area. Think of ATP or adenosine triphosphate as the cell battery power or energy source. Primary transport uses ATP directly, like pedaling a bicycle using your own energy. Meanwhile, secondary transport uses stored energy from another movement, like a father pulling a wagon with his child riding. The wagon moves because of the energy from the father who is pulling. Let's move on to the other one, vesicular transport. Vesicular transport is used for very large materials. The cell uses vesicles, tiny bubble-like sacs, to carry these materials. The vesicle is basically a small bubble made of phospholipid bilayer, just like the cell membrane itself. There are two types of vesicular transport, endocytosis and exocytosis. Endocytosis is when the cell takes substances inside. On the other hand, exocytosis is when the cell pushes substances outside. Three types of endocytosis are phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis means cell eating, where the cell surrounds and swallows large particles. It's like a person eating food. The food enters the body just as large particles enter the cell and are brought inside for processing. Pinocytosis means cell drinking, where the cell takes in liquid droplets and dissolves substances. It's like using a straw to sip juice. The liquid is taken in just as the cell takes in fluid and dissolves substances. Receptor-mediated endocytosis is when the cell takes in specific substances using receptors on its membrane. It's like a claw machine. The claw only picks up the toy it successfully grabs, not everything inside. In the same way, the cell only takes in substances that fit and bind to the receptors. Exocytosis is when the cell uses vesicles to push substances out of the cell. It's like squeezing toothpaste out of a tube. The toothpaste moves from inside to outside, similar to how substances leave the cell. So, Vesicular transport helps move large materials that cannot simply pass through the cell membrane. The environment surrounding a cell affects how water moves across the membrane. To understand this better, let's see how cells respond to a solution concentrations. Before we begin to this part, the solution we are referring to here is a liquid with many substances dissolved in it, such as ions, glucose, amino acids, proteins, and other molecules. In this discussion, we will use salt as example to represent these dissolved substances. Salt dissolves in water inside cells and breaks into very tiny particles or ions, which are so small that they cannot even be seen under a light microscope as separate solid particles. And we're also not talking about things like cleaning solution or contact lens solution. In here, a solution simply means a liquid with something dissolved in it. In this visual presentation, you can see bits of salts, but I only drew them as a representation. In reality, inside the solution, there are no visible salts like that because they are completely dissolved. Now, a hypertonic solution means there's more salt or other molecules like sugar dissolved outside the cell than inside the cell. For example, when making marinated beef, we often soak the meat in a salty marinade. In this case, the salty marinade is a hypertonic solution because it contains more salt than the inside of the meat. Because of this, water moves out of the cell through osmosis. As a result, the cell shrinks and becomes dehydrated. If you're thinking, why is this important? This explains how salt can preserve food. When water leaves cells, many microorganisms have a harder time growing, which helps food last longer. Without this process, salty foods would not lose as much water and would spoil more quickly. In animal cells, this is called crenation. Crenation means the cell shrinks and becomes wrinkled because it's loose water. Crenation is not about pets or animals we see in daily life. It is not something we can see with the naked eyes. It is about animal cells, like red blood cells, and it is usually seen under a microscope in laboratories. Why is this important? Scientists study crenation to understand how red blood cells react when they lose water. This is important because red blood cells carry oxygen, and if they shrink, they cannot function properly. It is also important in medicine and treatment. Doctors need to make sure that fluids given in hospitals like IV fluids are balanced so that blood cells do not shrink or get damaged. This keeps patients safe and supports proper treatment and recovery. Without this knowledge, scientists and doctors would not fully understand how cells respond to fluid imbalance in the body. Fun fact, crenation sounds cremation, but they are completely different. Cremation is the process of burning a dead body, while crenation is simply the shrinking of a cell due to water loss. Meanwhile, in plant cells, this is called plasmolysis, which means the inside of the cells pulls away from the cell wall because water leaves the cell. Plasmolysis can be observed under a microscope, especially in lab experiments where scientists use salt solutions to clearly see how plant cells lose water. Plasmolysis happens when plant cells lose too much water, causing the cell membrane and cytoplasm to shrink away from the rigid cell wall. So, if the outside environment is too salty, water leaves the cell through osmosis. Why is this important? Even though people don't usually put salt directly on plants, this can still happen in real life. For example, 1. Salty soil near oceans due to sea spray. 2. Over-fertilized soil where too much dissolved minerals act like a salt effect. 3. Poor irrigation water in high salt content. These conditions can cause plants to lose water from their cells, making them wilt and become weak. Plants need water to stay firm and upright. When plasmolysis happens, the plants become weak and wilted because its cells lose water pressure. This helps scientists understand why plants dry out in very salty soil or during lack of water. It is also important in agriculture because farmers need to make sure soil conditions are not too salty or plants will not grow properly. Without this process, we would not understand why plants dry out or struggle to form in certain environments. Now, let's look at the opposite situation. A hypotonic solution means there's less salt or sugar outside the cell and more water outside. Remember, a solution is simply a liquid with something dissolved in it. Because of this, water moves into the cell through osmosis. As a result, the cell swells. In animal cells, too much water can cause the cell to burst. This is called lysis, which means the cell breaks open because it has taken in too much water. When a cell bursts, it is usually damaged beyond repair and dies because its contents leaks out. Why is this important? Scientists study lysis under a microscope to understand what happens when cells take in too much water. This helps doctors make sure medical fluids and treatments are balanced so cells stay healthy and do not get damaged. Without this knowledge, cells could become harmed by taking in too much water. Meanwhile, in plant cells, the cell becomes turgid, which means firm, stiff, and full of water. Unlike animal cells, plant cells usually do not burst because they have a strong cell wall that helps support and protect them. This is actually a good thing because it helps plants stay upright and healthy. Think of a fresh lettuce leaf or a healthy plant after being watered. It looks firm because its cells are full of water. So unlike a hypertonic solution where water leaves the cell, a hypotonic solution causes water to enter the cell. Now let's move to the balanced condition called an isotonic solution. An isotonic solution means the cell inside and outside of the cell have equal solute concentration. Because of this, water moves in and out of the cell at equal rates. This is called dynamic equilibrium, where water is still moving across the membrane, but there is no overall net change. As a result, animal cells, or cells in general, stay normal. Meanwhile, plant cells become flaccid, meaning soft and not fully firm. Why is this important? Scientists and doctors often try to keep cells in an isotonic environment because cells function best when water and solute levels are balanced. For example, many IV fluids used in hospitals are designed to be close to isotonic so that cells do not shrink or swell. Without this balance, cells could lose too much water and shrink, or gain too much water and swell. So, in an isotonic solution, the cell stays stable and functions normally. To summarize, hypertonic is water moves out and cells shrink. Hypotonic, water moves in and cell swells. Isotonic, no net water movement and cell stays balanced. Another thing to remember is that diffusion has its limits. Let's see what those are. Diffusion works faster over short distances. Like when you smell food right in front of you, you notice it immediately. But if you are far away, it takes longer to smell it. That is why cells stay small, because smaller cells make it easier and faster for substances to move in and out. In multicellular organisms, the circulatory system helps transport materials quickly to different cells, like delivery trucks bringing supplies to different houses. So small cells and blood flow help transport happen efficiently. Understanding why cells stay small helps scientists understand how living things grow and why cells have size limits. Next, let's talk about what affects diffusion speed. Many factors affect the speed of diffusion. Shorter distance makes diffusion faster, like hearing someone clearly when they are near but not when they are far away. Smaller molecules move faster, like dust particles floating easily in air compared to larger objects. Higher temperature also increases speed because particles move more actively, like how people move more actively when it's hot compared to when it's cold. A bigger concentration difference speeds up diffusion, like people quickly spreading out when one area of a room is crowded and another is empty to balance the space. A larger membrane surface area allows more movement, like a bigger window letting in more air than a small one. So these factors can make diffusion faster or slower. Scientists study these factors to understand how quickly substances move through cells, which is useful when developing medicines and studying how the body works. Here's a quick summary of the main points on membrane transport. In summary, the cell membrane controls what enters and leaves the cell. Passive transport does not use energy, meanwhile, active transport needs energy. All these processes happen in all cells human, animal, plant, protists, single celled organisms like amoeba, yeast, and mushrooms, helping all living cells stay balanced, healthy, and alive. And that is how transport across the cell membrane works. I hope you gained a lot in this video. Please like, subscribe, and comment, and wait for the next week's upload.